guys, welcome or welcome back to my channel. Today we are going to discuss another topic from cell biology, that is intrinsic pathway of apoptosis. So I hope you might have gone through the previous videos related to apoptosis. If not, that I would recommend to firstly go through that, otherwise the topic won't be clear to you. So what is the name intrinsic itself suggests that the apoptosis is mediated by an intracellular signal such as a cell damage. And how does cell damage occur? Because of DNA damage, right? Okay, so firstly I need to discuss that in intrinsic pathway we need to understand the major role is played by the BCL2 family proteins, right? It is divided into two parts: pro-apoptotic and anti-apoptotic, right? Pro-apoptotic is the one that regulates apoptosis, and anti, as the name suggests, it down regulates or is against apoptosis or stops the apoptosis, against apoptosis, right? Pro-apoptotic is further divided into two families: the effector BCL2 family and the BH3 only proteins, right? So if we discuss about the examples, in effector BCL2 we have BAX and BAC. In BH3 only we have BAD, BID, Puma, Noxa. And whereas in anti-apoptotic we have BCL2, BCL, XL and BCLB proteins, right? What happens whenever there is an apoptotic signal by a cell damage, apoptotic stimulus by a cell damage, thus it activates the BH3 family, and this family stops this anti-apoptotic and activates or promotes effector BCL2 family, which helps in the formation of MOMP. We will discuss what is MOMP, but you need to understand that BH3 family inhibits anti-apoptotic only when the apoptotic stimulus is there. Otherwise anti-apoptotic is active, because रेगुलर बेसिस पे एपोप्टोसिस के नॉट बी मीडिएटेड ना. अदरवाइज अगर ये लोग इनहिबिटेड रहेंगे तो हर टाइम ही एपोप्टोसिस होता रहेगा सेल के लिए, फिर तो ज़्यादा डैमेज हो जाएगा, राइट? So I was telling about MOMP. MOMP stands for mitochondrial outer membrane pore. So we have a very great role of mitochondria in the intrinsic pathway, right? This is the mitochondria structure. This is the outer layer and this is the inner layer. This is the intermembrane space. So इस intermembrane space में we have so much of cytochrome C produced by oxidative phosphorylation, right? So this cytochrome C has to come out, because this only mediates or acts as the stimulus for the intrinsic pathway to be conducted. So here, this, as soon as we receive apoptotic stimulus, activation of BH3 family and activation of BCL2 family leads to activation of BAC and VAX, right? VAX remains stick to the outer membrane, right? So as soon as it gets activated, it leads to the formation of channel like MOMP, and this cytochrome C comes out, right? As soon as this cytochrome C comes out, it gets bound to APAF1. What is APAF? APAF stands for apoptotic protease activating factor, right? Cytochrome C does not bind to a single APAF, it binds to 7 APAFs, like right. So this is cytochrome C. Okay, as soon as it binds, this is known as apoptosome. This is heptameric structure, right? So as soon as it binds to it, the card present inside it becomes exposed, right? It becomes exposed. So this what is card here? I'll write the full form of card: caspase recruiting domain, right? So this caspase recruiting domain gets exposed, and thus it leads to the binding of procaspase here, right? This is procaspase 9. This procaspase gets activated and becomes caspase 9, which leads to activation of procaspase 3, and which activates and becomes caspase 3. Here procaspase 9 is the initiator one, whereas procaspase 3 is the executioner, right? As soon as the caspase 3 gets active, apoptosis of the cell occurs, right? So this was it. Major role is of this BCL2 family. I hope the topic might be clear to you. If you have any doubt, please drop down in the comment section. Please like, share and subscribe. Thank you.